Thanks for everybody for joining into our first virtual session. I wanted to thank Carmen for taking the time for doing this for Science Outreach. We really appreciate it. And I'd like to invite everyone to participate in some questions at the end of the session, if they have any. And I want to introduce Roland True, who is one of our Science Outreach volunteers, and he will be introducing Carmen.
Yes, good afternoon, folks, or good evening, rather. I'm glad you are willing to be our guinea pigs for our first virtual presentation. I'm really looking forward to get back to seeing real people, but for the time being, we have to do it that way. Before I introduce Carmen, I just want to remind people that we are recording this session. So if anybody feels that they don't want to be recorded, you can turn off your video. And while Carmen is talking, it would be good if you all mute yourself. You can do that by going on the mute button at the bottom left corner of your screen. Also, you might already click on the chat box, because in the chat box, that's later where you can actually type in your questions. At the end of the presentation of Carmen Allen, you will, of course, if you prefer, be able to ask your question verbally as well, if you would do that. Otherwise, just do it in writing in the chat box, and I will read the questions to Carmen at the end of her presentation. And then at the end of the presentation, you can also ask her any questions. There is an option to raise your hand. So if you go into the chat box, you should find that in the chat box, you can raise your hand. But I would really ask you to do that only towards the end of the presentation, so that Carmen can do her roughly 1 hour presentation, I guess. And then we have question time towards the end. So, and we hope everything will go right for this first presentation. It is my real pleasure to introduce Carmen Allen, my colleague who got her BSc from the University of Alberta in environmental biology. And she did her Masters of Science in Carleton University on mycological subject. Carmen has been Science Outreach volunteer for about 11 years now, participating and helping in various activities that we do, some organized by her, many organized by herself. Carmen is also a Athabasca University lab instructor, has been for about 9 years instructing several different labs, and she's also an academic expert. In January of this year, Carmen started her PhD with Toby Spribilli on lichen symbiosis at the University of Alberta. So that's what she is doing right now. And we are really looking forward to her presentation. Thank you, Carmen, for agreeing to do this. And the floor is yours. Thank you.
Awesome. All right. Thanks for the introduction, Roland. Today I'm going to be talking about the age of enlightenment, and I'm going to be talking about symbioses, and also what I put DNA sequencing can tell us about those organisms. This happened to me last time. Okay. So our topic for today, I'm just going to go over generally what we're going to be talking about. I'm not quite sure how long this is going to be, but here we go. So I'm going to start talking a little bit about what is a symbiosis, because this may be new to some of you guys. And I'm going to give a bunch of examples as well of different symbioses. I'll then talk more about DNA sequencing. So we'll talk about what DNA sequencing is. We'll talk about how DNA sequencing can help tell us who are the partners in a symbiosis, and also what are they doing. So we're going to start with what is symbiosis and talk about some examples. So symbiosis is when organisms live physically close together. And this term was first came about in 1879 by this gentleman, Henrik Anton de Berry, and he was a physician and also was a botanist and a mycologist. And he defined this as the living together of unlike organisms. And he came up with this term when he was looking at lichens. So this symbiosis term originally came from lichens. This is another different sort of definition that I found, probably on Wikipedia, of symbiosis, which is any type of a close and long-term biological interaction between two different biological organisms, which I would not agree, it's just two, be it mutualistic, commensalistic, or parasitic. So we're gonna go through what those mean as well. So there's a whole different range of the types of relationships that symbiosis can be. And one really handy thing in science and in biology in particular is there are lots of big fancy words, but often you can break them down into really meaningful parts that help you remember what the words are. So symbiosis, the word sim means together, and biosis means living. So that's why that's how that word came to be. And I'll do this throughout the talk a little bit with words. I don't know, I really like to break up words because that's how I remember what they are. Okay, so this is a like huge way oversimplification of the different kinds of symbioses. So it's way more complex, but this is a general idea. So if you have two organisms that are living really closely together and both are really happy and benefiting somehow, we call that a mutualism. So you recognize the word mutual. Ism means something that's direct. So they're having a direct benefit to each other. Another kind of symbiosis is commensalism. And this word comes from eating from the same dish. So com means together, and mensal comes originally from table or meal. So eating together, but they're not really impacting each other too much. So in this kind of relationship, one partner is kind of like meh, doesn't really matter. I think my kids would probably say whatevs, that's what they like to say, doesn't really care. And the other one is quite happy. So that's commensalism. And another kind of symbiosis is parasitism. And para means beside, and then this SIT part comes from sitos, which means wheat. So I'm not quite sure how they put those two words together, but some eating and then beside. So it's eating, benefiting somehow from another organism, and that organism is suffering. So that's what happens if we have a parasite, we're not as healthy. So those are three types of general symbioses. So I'm just gonna go through some examples of symbiosis that I found really interesting when I was researching for this topic, and I wanted to share with you. So these come from a huge range of environments and different groups of animals and microbes. So the first one is mutualism. So mutualism, remember, is when both are benefiting between sea anemone and clownfish. So this is quite like a popular way of presenting mutualism. And so a sea anemone, you see them on the ocean floor and they have these tentacles. And sea anemone tentacles have these really interesting organs where they actually have these miniature harpoons that can shoot out and help them to stun and capture prey. And so that's really interesting because the clownfish is able to live within these tentacles and they don't get stung somehow. It might be because they have a special mucus all over them, but they also are providing benefit for each other. The anemone gets, from these clownfish swimming around, it actually gets a lot of aeration, which gets more nutrients and stuff moving through tentacles. The anemone gets protection because these clownfish will actually chase off some of the predators that would eat an anemone. There are reports that the clownfish actually can help in hunting, so they can actually lure prey towards the anemone. And also, you'll hear this time and time again tonight, is that the poop of the fish is actually consumed by the anemone, so bringing nutrients to the anemone. The clownfish gets food scraps that are left over from the anemone feeding. They get protection because these stinging tentacles will sting other fish and other predators of this clownfish and will leave the clownfish alone. Also, these clownfish can raise their young within these tentacles and protect them. So it's a real benefit, and it's a long-lasting benefit that has been going on for a long time. So that's one example. One that you've probably heard of as well is corals. So we hear a lot about coral. This is the Great Barrier Reef here, an example, and how important this habitat is. And the structure of the reef is all created by these tiny colonial polyps. And they're related to the sea anemone, sort of in the same group of animals. So they kind of look like that, but these are teeny tiny. This is, I don't know, like 2 millimeters across. And then they go a little bit deep into the coral, and then they extrude from their bases calcium chloride, which makes up that hard reef. And these have a mutualism with many organisms, but the one that we mostly think about are these zooxanthellae, which are dinoflagellates, which is a microorganism that is able to take the sunlight and make that into energy. And so they can share that with these polyps. And then the polyps can provide a place for these zooxanthellae to live quite comfortably. And you've probably heard of coral bleaching, which is a real problem. And that happens when something in the environment changes. So maybe the pH of the water or the temperature of the water. And for some, I think, largely unknown reason, these polyps actually evict the zooxanthellae. And that's why they lose color, because this color is a lot derived from the dinoflagellates. So that's something that you might have heard about, is the coral reefs, and you may or may not know that it's actually quite an interesting symbiosis. And there's a lot more to it than what I've shown here, but that's just the very basics. I have to, okay, there we go. This one I actually just learned about, and I didn't really know about sloths and moths. This is just from a 2014 paper, and was just brought to my attention by one of our grade 7 teachers, who was saying, have you heard about the sloths? Because she teaches symbiosis. So this one is quite new to me. And this story came about because there are 3-toed and 2-toed sloths. And I believe it's the 3-toed sloths, for some reason, travel once a week. They climb down from their tree and poop on the floor of the forest. And why they don't just hang out in the tree and do that is a bit of a mystery, because every time they go down, it takes a lot of energy, and they're at risk of encountering predators on the floor of the forest floor. So these scientists here, Polly and others, they didn't know why the sloths would bother doing this. So they went to find out why. And they found out by sampling their fur and sampling the poop that was on the ground. They came upon this hypothesis here that the sloths actually have within their fur, they have this really robust algal community. So they have all these algae living in their fur, and they have, it's not really shown well here, they actually have these kind of structures in their hairs that allow the algae to grow in their hair. They also have this amazing moth population in their fur, which I did not know about. So all of these are part of this story. What happens is that the sloth climbs down the tree to defecate, and these moths that are living in the fur, they come down with the sloth in its fur, and then the female sloths that actually that are ready to lay eggs, lay their eggs in that poop. And then the larvae of the moth actually eats that poop and becomes an adult in that poop, and then emerges, and they will then go up as adults. They can fly, so they'll go and they'll find a sloth and make their way into the fur again. Meanwhile, they're bringing nutrients, and they're not sure how exactly, these scientists, how they're bringing nutrients, if they're bringing actually little pieces of nutrients from this dung and into the fur, or if they are somehow associating with other fungi that are releasing minerals, like these ones here, into the algae. So they're actually feeding the algae, all these moths. And then the sloths are actually kind of grooming themselves and taking the algae off of their fur and eating it. And it's a really good source of nutrients. So this is a new one to me. It's really interesting. And I thought that you guys might find it kind of cool. Okay. One that you might have heard of as well is the human microbiota. And I know that Shauna Zenteno did a whole talk dedicated to the human microbiota maybe a couple of years ago for Science Outreach. So our microbes all over the surface of our body and inside our body is an emerging field to figure out what these microbes are actually doing in and on our body. This is just a popular book, a pop science kind of that I read a while ago, which is sort of the idea of how we've over-sanitized our kids over the years, and that has kind of led to some problems. So this field is really interesting to me, and there's a lot of implications of the microbes that live all in and on us, to do with things like asthma, obesity, and allergies, and different immunological conditions. So it's quite interesting. And since Shauna gave a whole talk on that, I'm not going to talk too much about it, but it's quite interesting. Okay, another mutualism that I wanted to talk about are mycorrhizal fungi. And these fungi are really interesting. And most, if not all, plants that you encounter in the boreal forest around Athabasca or wherever you are, they are associated with this underground network of fungi. And you can't see them very well. They're just made of these tiny filaments, which are the cells of the fungi. But they're really important to how plants grow and how nutrients are distributed between plants and underground and then moved above ground. So this is a schematic that I found, and it's showing how this symbiosis works. This is a tree that's photosynthesizing, so it's grabbing sunlight and creating sugars from that. These are then able to travel down the tree or plant or whichever into this network here, and then can either become part of the fungi, or it can actually go a number of places. I'll talk about this little plant here in a little bit. So the fungus is benefiting because it's getting all these photosynthates from the plant, and then the fungus is mobilizing all sorts of nutrients in the soil that the plant can't get at by itself because it doesn't have the right ways of breaking it down. So those are able to travel through this mycorrhizal network back into the tree. So that's kind of the basic how this association works. These were really interesting, the mycorrhizal associations, because you can have all sorts of things connected. You can have trees of the same species connected to one another and actually sharing nutrients. You can have other plants that are actually tapping into this network but not helping at all. So they're not providing anything, they're just taking. So for example, you may have seen this Monotropa uniflora around in the boreal, and it is, some people call it ghost pipe, and it is not green because it doesn't have to be. It does not photosynthesize. It is taking, it's kind of like this plant right here, it's tapping into this underground network and basically parasitizing other plants and taking their food. So this is a little bit of a complicated relationship, but it's really cool. I think, yes, and I have, so there's a couple of different kinds of, well, there's more than just these, but here's a couple of kinds of mycorrhizal fungi. These ones are called endomycorrhizal fungi. Endo means within. So these they're called arbuscules that look like little trees, and they're actually inside the cells of a plant and really directly interacting with the plant from within cells. There's a whole other group called ectomycorrhizal fungi. So they're outside. They do kind of work their way in between cells, but they don't actually penetrate into the cortical cells. They're just on the outside and interacting that way. Okay, the next mutualism that I'll talk about is lichen. And this is what the lab that I'm in is studying. So I just wanted to show a number of different kinds of lichens. This is a hair lichen, so it's hanging from a tree. And you'll see these all around here. We have other ones called foliose lichens, so they kind of look like leaves. And these can be on wood or on the ground. These kinds here are kind of the most simple lichen form, and these are called crust lichens. So sometimes you may see rocks, and it just looks like a coloration on the rock, right? Like an orange coloring or white or black or a whole bunch of different colors. And that's the most simple form, and those are also lichens, the crust lichens. You can also find these beautiful fruticose types of lichens where there's projections, and sometimes there's these really beautiful colors that you can find. I find these often kind of look like golf tees in the forest if you see them. Okay, so the lichen symbiosis, this is sort of the textbook definition of a lichen. If you've learned about the different partners in lichen symbiosis, there's the fungal partner. So these are these sort of filamentous cells that make up fungi. And then there's some sort of a photosynthetic partner, and it can be a green algal species, or it could be, you could either have both. It could also be cyanobacteria. So there are some different options here of the kind of partner that's able to harvest energy from the sun and turn that into some sort of product that can then be passed to the fungus. The fungus is providing a nice sort of structure to house the photosynthetic partner. So without that, these may desiccate and not be able to survive for very long without the fungal partner. So this is just a little bit more of what a lichen would look like. This is growing on birch, and lichens they have this cortex here that's on the outside, and it is also right over here. And this is kind of the part of the lichen that you often would see. It's giving it a lot of structure. Below that, this is where you'll find the photosynthetic partner, is all these algal cells right below that. So they have to be close enough to the surface to be able to catch that sunlight. So those are the algae. And then there's this loose sort of spaghetti of fungal hyphae in the middle. And that's the main fungal partner in here. So I'll talk about this more recently, or closer to the end of the talk, but there's also more partners as well that have been found to be associated with the lichens. There's yeasts, and also lots of bacteria. So this is an emerging field that I'll talk about closer to the end when we're talking about some of these sequencing techniques. Okay, so those were all mutualisms. Now I'm going to talk about, I think this is my only slide, of commensalism. So commensalism is when one partner doesn't really care, is not impacted, and then the other one gets some sort of a benefit. So often when you hear commensalism, you might hear about commensal bacteria, like this is an example of commensal bacteria. But I don't quite believe that any sort of commensal bacteria are actually doing nothing. I think they're usually probably giving us a benefit of some sort, even if it's just being there so that some other microbe that isn't as good would colonize that space. So that's often one way you might hear of commensal bacteria. And then when I was reading up on this, two that I didn't expect to come across are dogs and cats. So originally dogs evolved from wolves, and then the idea about that is that these wolf packs kind of became more comfortable around these human settlements, and then they would maybe maybe eat some of the garbage that was left around, but you know, it wasn't impacting the humans really because they weren't harming them. And that's kind of the start to how that association started between, and the domestication of dogs. Cats, I read that cats actually, they think were fully commensal before they were domesticated. So they were in some areas like fully reliant on humans and fully reliant on, for example, the rodents that come with human settlement before they were actually brought in as pets to our homes. So I thought that was kind of interesting. Oh, right, I have this example too of Remora fish. So these Remora fish have these amazing structures. This is actually a dorsal fin adaptation. So dorsal fin is like, you know, in Jaws, how there's that dorsal fin on the back. So this is actually on the top of the fish. I thought this was a mouth part at first, but it's not, it's on the top. It has these ridges that when the fish swims in one direction, it'll actually help it stick to the whatever it's trying to stick to. It can be a fish, it can be a boat, it can, so these are some pictures of these Remora fish hitching a ride on a nurse shark. So there's presumably, I don't know, this might be debatable, that the shark is not impacted, not sure. And these guys get a ride, so they're benefiting by that. One thing I read about these, which I thought was fascinating, is that in the Indian Ocean, people actually use these Remora fish to fish. So they'll, like, I don't know exactly, they'll tie maybe a rope onto the end of the fish, throw it out there to the Remora fish, and it will go after turtles. Apparently they like to hang on to turtles. And then the fishers will pull in the Remora fish with the turtle. And then it's kind of brutal, but it's really fascinating that I had no idea you could do that. Okay, so I will give a couple of examples of parasitism. And just a couple, I feel like there's so many cool examples, but I want to stick with the good news, so I won't cover too many parasites. But this is one that you've probably heard about, is Whirling disease. And this is a disease of salmonoid fish. And it is due to this critter here, Myxobolus cerebelis, which is also in the same group that is a part of corals and also anemones. So there's in the same group of animals. This one's just really tiny. And this one makes its way into I believe it makes its way into the brain of these salmonoid fish. It deforms them and they swim all funny, which is why it's called whirling disease, because they aren't swimming properly. And it's getting to be a pretty big problem in Alberta. So that is one parasite. And remember, parasites, they're generally not killing the animal or whatever it's parasitizing, but it's definitely weakening them and taking resources from that host. The other one that I wanted to talk to you about, which I've always thought these are so cool, are parasitoids. So I'm actually not sure when I put this up if this is a real true symbiosis, because these actually kill their hosts. So this here on the right is a wasp this is called an ovipositor right here, and it's actually going through the bark of a tree and into some sort of a larva that is living in the bark. And then it'll pump its eggs into that larva, and then the eggs will develop inside as the larva is alive, and then it'll burst forth like this one here came out of. This looks like a caterpillar. So they're quite brutal. If you've seen the movie Alien, when there's like aliens coming out of human abdomens, this is the same idea, but with insects. So these are kind of cool, parasitoids. The reason why they're not parasites, parasitoids, is because they actually will kill their host. Okay, so Originally, I gave you a very simple view of what the kinds of relationships are that there can be in symbiosis, and in reality, it's complicated. There's lots going on, and we can't really narrow it down to just those 3 types that I gave you. There can be lots of different partners. The status can shift over time, so something that was a mutualism could become a parasitism, or a parasitism could become a mutualism. So this might be based on what's available at the time. So if you have a mutualism and then for some reason there's whatever that symbiote is providing to you, there's just enough of it in the environment. Like if you're a plant and you're getting fertilized with exactly everything that you need, well, you might not want to actually, you don't need your mycorrhizal partners anymore, perhaps. So it's actually, they're becoming a parasite because they're only taking, and you don't really need to, so it can shift and it's a little bit complicated. I just put this example here because these are little organelles that are inside of human cells and lots of different, any animal that you would see. And this little organelle here, it's hypothesized, or they think that it might have actually been a parasite that was in these cells to begin with. And somehow it shifted to actually being a symbiosis where it was actually providing energy to our cells. And then eventually these little parts of our cells lost so much of it that it couldn't even function on its own anymore. It had to remain in our cells. So this is how it can sometimes shift over time, and it's not always straightforward. Okay, so that covered what is symbiosis and some examples. So now I'm going to talk about what is DNA sequencing. So this is just a very brief sort of background of what we call the central dogma of molecular biology. So the word dogma it means that it's even more than like a theory or like a hypothesis. This is something that in molecular biology we firmly think is true and base a lot of science around it. So that's why it's a dogma. And the dogma is that we have DNA in our cells and in any organism's cells. This is what is inherited and passed on to kids. That is then made into this single-stranded type of molecule called RNA. This has a little M in front of it because it's a messenger RNA. And then those are made into proteins. And you've probably heard of the genetic code. And what that means is that there are these are called bases, but we always just write them as these letters. There's only 4 of them. There's A, T, G, and C. And they always pair up with each other. A's and T's always go together, and G's and C's always go together. And this molecule down here has U's instead of T's, but that's fine. And then the reason why it's a code is that every triplet here means a different amino acid in a protein. So that's the central dogma. And then these proteins are what make our body run. So just to go over that a little more, DNA is what you're passing on, right? So it's the hereditary material. When we take all of the DNA in an organism, we call that the genome. So you might have heard of the human genome. So that would be all of the DNA in a human. And we have chromosomes. This is a plant cell, but, I think these are plant cells. And so we have chromosomes, just like those ones, but then bacteria, the genome is a lot smaller and is just a circular genome. This is an example of a virus, a circular or a coiled viral genome. This one's actually SARS-CoV-2 genome, so that's what they've figured out so far. It's an RNA genome. It's one of the bigger virus genomes around, apparently. So RNA, that's this one here, this single strand. And the reason why there's RNA is because the DNA is often in cells like animals and plants. The DNA is in this little nucleus of the cell. It has to get out somehow into this part where the proteins are made. So this messages are able to get out. So that's why we need the messenger RNA. And then that's then made into protein. And proteins do all sorts of cool things. There's proteins here that make energy for our cells. There's proteins that make other molecules. This one's making messenger RNA. There's messengers that proteins that sense our environment and make changes in our cells. There's different proteins that let things in and out of our cells. So these are transporters. This is one that we're all interested in right now. This is an antibody. And we're all hoping to get some COVID-19 antibodies from some vaccinations soon. And then these are some proteins that hold cells together. So they are have a lot of different kinds of roles. And our DNA is a little bit different between individuals of a species, and then a little bit more different between different species. So that can make these proteins a little bit unique, and that's what makes all of us unique for the large part. Okay, so what is a DNA sequence? So I talked about those bases, the letters, and a sequence is basically just stringing together all those bases into a long file. And it just depends how much you can, if you had a whole genome in there, it might be a really, really large file. So this is just an example of one that I took from my computer of a bacterial gene. Okay, so I wanted to talk a little bit about how we actually how we go from here, a molecule, and digitize this information into letters on a screen. So the first way that scientists were able to sequence DNA was through something called Sanger sequencing that was developed by Frederick Sanger. Was first commercialized in 1986. And the way, just briefly, that this works is that it is done through synthesizing DNA. So you'll have one strand of DNA, and we have special reactions that we can use to copy that DNA. And these little T's here are little nucleotides that are coming in to copy the DNA, and they're all specific to its match. And then what Sanger figured out is that he had these other kinds of nucleotides that would just stop whenever they were added. And so he would basically have, at the very end, all of these sequenced, or these pieces of DNA that were all different sizes. And then you would know which nucleotide stopped at each time, and then you could separate it out in this, they put it in this capillary gel and pass it through. And the little guys go faster than the big guys because they can't move quite as easy. And you get this series of peaks that it reads the light, and you can know what the sequence is. So with this technology, you can do about 100 to 1,000 base pairs at a time. The first human genome was done using this. It took 15 years, and it cost more than $1 million to do the first human genome. So this was a huge deal. And one of the disadvantages of this technique is you need to know something about your DNA to get the process started. So you can't just take something completely unknown and do this kind of sequencing, which is a problem if you don't actually know what you're looking for. So what we're doing a lot of these days is now called second generation or next generation sequencing. And in this technique, you're basically taking an entire genome and you're cutting it up into tiny little pieces. So these are all the little cuts here. And then all those little cut up pieces of DNA are being linked onto this tiny little slide. And then as we're doing that same kind of copying reaction where you're adding nucleotides, each nucleotide has a fluorescent label to it. So whenever it's added, it will kind of shine a certain color. And each of those 4 bases, the 4 letters, has a different color. So you'll get this kind of colors shining, and that each one will correspond to a different base. And then you can just, the computer will see the change. This one maybe went from green to red, and that would mean TA, and it would do each color. And then because there are so many, you can do this so many times that even though you've cut up your whole genome into little tiny pieces, if you get enough pieces, then you've covered everything. So it kind of might look like this, all these little pieces. And getting these little pieces into an assembled sequence that is just one long string is a whole discipline of science called bioinformatics. So this is a really big deal, and these days is trying to take all this massive amount of data that we get from things like sequencing and turning it into something that makes sense. So there's millions to billions of these tiny little fragments that are sequenced at the same time. So that's why it's high throughput sequencing. Using this technique, the human genome was sequenced at 2 months instead of 15 years. And the cost was just a fraction of the cost. So I'm trying to think, I have a video, but I'm not going to show it because we don't have that much time. Okay, so now there's actually a third generation of sequencing that is up and coming, and there are a number of different strategies. This isn't really widely used quite yet because there are lots of errors. It's not quite as good that way as the second generation sequencing, but there's a couple of ways. Well, there's more than two, but one of the ways that one company is doing is that one piece of DNA, so there's no amplifying like I was talking about earlier, passes through this little tiny pore, and each nucleotide is taken off, and each time one is taken off, it is somehow they can discriminate between the different bases, and then they can record that. And this is another one where there's one molecule that's being synthesized, and then there's a light that is emitted when each base is synthesized. And so you can actually do a really long molecule using this technique and not all these little fragments. So this is kind of this will be more prevalent in the future, but it's not quite there. Another advantage of this technique is that it doesn't take a big instrument. And this is something that's as big as a flash drive that you can actually use to do this third generation sequencing. Okay, so one of the interesting problems of working with symbiosis is that you often end up with a mixture of genomes. So here I've just given some examples of circular bacterial ones, viral one, and something that looks like a chromosome. Because they're symbionts, sometimes you can't separate them because they won't be able to survive without the other. So we actually have to figure out a way to sequence all of these things at once and somehow recreate these genomes to understand what it is. So the way that we do that is that these are all of these pieces are broken up. So this is a pool of these fragmented DNA pieces. And then this here is actually many steps, but I've simplified it a ton, where all these pieces are characterized, we call them into bins. So there may be some different characteristics between, like, let's say a viral piece of DNA and a bacterial one versus maybe a fungal one or an animal type of DNA. And so those are kind of signatures that computationally we can pull out. And so these are actually put into bins. And so each bin would theoretically be a different species. And then using bioinformatics, these are then stitched together into one sequence, and we call that assembly. So this field is called metagenomics. And genomics is, the gen part means genes. So genes is a segment of DNA. For the purposes of this talk, I'm going to say that it's generally a segment of DNA that makes a protein. So all those different proteins are made by genes. Omics means the study of the totality of something. And then meta is a level above or beyond. So this is where the mixture comes in. So you're not dealing with just one species and trying to sequence it, you're dealing with many of them. So it adds a whole level of bioinformatics to your projects when you're working on metagenomics. Okay, so I'm going to talk about a couple of, I'm going to talk about how, just a little bit of background on how can we actually tell who's in this symbiotic relationship. So if your symbiosis is between a human and a dog, that's pretty easy. You know what a dog looks like, you know what a human looks like, and you know what they're related to. Let's say you're trying to understand this symbiosis over here. And there's not a whole lot to go on. This looks like, sausages and Timbits. So it's not so easy. We don't really have a lot to go on in terms of identifying these. Just a little background in how we actually figure out how things are related to each other, just a little bit of history. This is Darwin, who made the first tree of life, or trying to understand how to conceptualize how things are related to each other. So this is a really famous first crack at how species are made. And he writes, I think, up here. This is 30 years later by Ernst Haeckel, who made a little more of a elaborate tree, and it actually looks like a tree. And we have plants, animals, and protista, which basically means everything else that doesn't belong into the other two. This is a way more modern tree of life. This is from 2016. We are somewhere over here. I'm not even sure where. This is like all of plants and animals right here. The rest of this is bacteria, so there's a lot of diversity there. And this is a whole other group called Archaea. So if we're looking at dogs and people, we might look at traits like teeth and bones and different things like that. We call that morphology to tell species apart. If we're just dealing with these tiny microorganisms, especially if we can't grow them, so we can't really tell what it likes for nutrition or anything like that to characterize it, we use DNA sequencing. And so this is an example of a bunch of DNA sequences of bacteria that I put together, and they are what we call aligned. So we've used different tools to try to match these up as best as we can. And there you can see that these are all sorts of different bacteria. There are parts of it that are really similar, like right here, all of these are very similar, and then parts where it's really different. So we can use a mixture of these similar and different parts to get an idea of how all of these different species are related to each other. This is just an example of some relationships that I put together. So these are based on those data. This is what I got. And the details don't matter at all, but you can see that you can kind of get an idea of which species are more related to each other. If they're really close together, then they're probably really close to each other. These are the same ones, just presented two different ways. So I'm just gonna finish up with two examples of where sequencing data was used to understand symbiosis. So this is one that I just read about. It's a 2020 paper. And this is a symbiosis between nano-haloarchaeon and haloarchaeon. So I showed this image earlier. These are really interesting organisms. They aren't bacteria. They aren't, they don't have cells like us where there's a nucleus and stuff like that. These ones are really extreme organisms. In this case, these ones are salt-loving. They were actually pulled out of an Italian artisan, what do they call it? I forget the fancy word for it, where they make salt. Saltern, anyway. So they ended up sequencing using metagenomics because they couldn't quite separate them all the way. And they built their own tree. So that's one of the kinds of things they would use with this data. They were able to figure out what the relatives were, and that might tell you what kind of characteristics that species would have. This one, the nano halobium is the little one that looks like little timbits here. They also can take this sequencing data and they can actually look for interesting genes in it and know what kind of genes that it has. And so one of the things that you can look for if you get the sequences are genes for maybe breaking down different substrates for food. So with this genome information, they found out that this is the nano one, the small one, and this down here is just a little bit of the big cell. So these are different species. And they were able to find that this nano halo archaeum, it is missing all sorts of things. It can't make its own amino acid, which is the building block for proteins. It can't make lipids, nucleotides. This is what makes up DNA, is nucleotides. So this organism cannot live without its partner because it's just missing so much. But these are really an interesting pair because the halomicrobium, it's able to live off of chitin, and chitin is what makes up your fingernails and hair, and also insect exoskeletons, and it's able to live off of chitin, which is a complex sugar. But the nano halomicrobium is not able to. It is able to live off of other kinds of sugars called alpha D-glucans. So these two organisms together are able to digest different kinds of sugar and pass them on to each other and share them. So it really allows them to live in diverse and changing environments, because if they have chitin available, they can use that. If they have something else, then they can use something different that in that environment. So it really helps them to be agile and use different substrates to live off of. This is one from Toby Scribbala, who's my supervisor right now at the University of Alberta. This is from 2016, and there are a whole bunch of other authors also on this paper. And this one is really interesting because they were able to uncover, using sequencing, a hidden yeast in lichens. So they were really curious because there are two different lichens. They're both the same genus of fungus called Bryoria. But they also had the same algae, but they look different and they didn't know why. And so they started sequencing this. They were actually sequencing the RNA, not the DNA, but that's okay for this talk. And they found, oddly enough, that there were these extra yeasts in there that nobody had ever found before. And that kind of correlated also with the way that they looked different, because nothing else is really different. Even though they have different species names, they aren't really different in any other way, and no one really knew why. So they were able to pull out the information from the transcripts and label that yeast using really special microscopy techniques. And so then these little green blobs are the yeast. And you can see there's more here than over here. So one of the conclusions of this paper is perhaps this yeast was leading to these different types of lichens. So this is, just to show that, yeah, they did the same kind of thing where they made some alignments and they created a tree, and they found out that it was this Cyphobasidium, and they knew it was, related. It was in the Basidiomycota, and it was related to these other types of fungi. And then this is a really cool video I wanted to play for you. They were able to label the three major parts of these lichens, and I will play it. This is way better than the image that's in the paper, because you can see that these yeasts are the green splotches near the outside is the yeast. So it's near the top. The red ones here, this red here is the algae, and these purple lines, that's the main fungal partner that's below that. So it's quite striking. They show you a bunch of different angles. And they basically just, to do this, they took a whole bunch of sections and then used computation to make them into this 3D movie, which is really cool. Okay. Oh, okay, there we go. Okay, so back to our topic for today. We've covered it all, which is good. It's about the right time. So I'm gonna leave you with a final thought that I found when I was reading about this, that you've probably heard of survival of the fittest, which is sort of Darwinian evolutionary theory. And I came across this quote and I really appreciated it by Lynn Margulis. She was really important to bringing awareness to something called the endosymbiotic theory, which is I talked about earlier about some of our organelles coming from symbionts that are living inside of cells. And she had this quote that life did not take over the globe by combat, but by networking. So I thought that was a really interesting way of looking at evolution, as opposed to sort of a Hunger Games, whoever survives wins kind of way of thinking about it. Okay, I wanted to say thank you to the University of Alberta, where I'm doing my schooling. Also at Athabasca, I'm working here in the lab to do some of my PhD work, so that's really amazing. And they've helped to fund some of the courses that I've taken, like workshops, not the university courses, through some PD funds, so that's been really great. I wanted to acknowledge NSERC, who is funding part of my research and is also funding some of the outreach activities that we have going on in Athabasca with the schools. And thanks to Science Outreach Athabasca for inviting me to talk. And I wanted to thank Alejandro Huerca Delgado for some of the photos of lichens. And I'd be happy to take any questions.
Thank you, Carmen, for your presentation. And the way we want to handle it now, so people who have questions can either write it down in the chat box, if you go to the chat box at the bottom of your screen, or if you want to ask the question directly, please do so. We have the raise hand item, but you might as well just jump in and ask the question. So I would clap if I knew how to do that. There's a way to do that somewhere, but I'm not familiar enough with that new software yet. Anyway, thank you, and I'm waiting for people's questions. And at the end, before you leave, I believe that Linda also might have a few comments and announcements. She wants to know how many people there were. So if you're not alone on your screen, Linda wants to know how many there were. I see the first question or comment here. The vacant form of vitamin D supplements comes from lichens. Have you come across this in your research?
Yeah, Karina, I didn't know that. Tell me more. Would you know what kind of lichens and anything like that? I don't know. I have never come.
Hi.
Hi, Karina.
Hi. No, I don't know, but I just know on the back of the supplement bottle, it just says that it's from lichens. So I was just always curious how that worked, or how they extracted it from lichens. And yeah,
I wonder what vitamin, I mean, there are lots of novel compounds that people are mining for interesting compounds in many different organisms. I know Roland's done a bit of that as well in his research. So lichens, definitely they've looked for lots of different compounds that are medically interesting and useful. I haven't heard of that one specifically. So vitamin D, I'm not sure why it would be there.
Oh, but, okay.
I don't know, but that's really, when next time I'm in your store, I'll come in and look at the at the packaging.
Yeah, specifically the AOR brand has a vegan source, and that's what it is, because all the other sources are from the lanolin in sheepskin, and then the vegan one is the lichen. So yeah, talk about it more.
AOR brand. I wrote it down.
Yeah.
And we got some information in the chat box from Jean, or there's a link actually. So this is something to look up.
Yeah.
That was from Jean.
Thanks for Jean.
That was from Jean Préau, and you notice Google is your friend?
Yes, thank you. So what does it say? I don't want to, I'm worried I'm going to click on it, I'll lose the meeting or something crazy.
No, that's just for later, if you want to.
Okay, for later, okay.
Most of the other ones were ads from Amazon or places that were selling that variety of vitamin D, and that one is more of a scientific one.
Awesome. I have a private message from Kim. What would you like high school students to know about lichens? That they're really important to lots of different ecosystems, and that, you know, you hear about caribou who are often in trouble, and they feed off of lichens quite a bit. They're in basically every habitat. They are really prevalent, and they're also really interesting because they are one of the first colonizers. So if you see a bare rock, they're able to actually to build soil eventually on that rock, which is really interesting for taking bare ground and making eventually a forest or grassland or whatever out of it. So that's something that I think high school students should know about lichens.
More questions? I'm looking at the screen here, but you can also raise your voice if you prefer that. And maybe while we wait for that, Linda, why don't you kind of wrap things up while we wait for possibly more questions? You might have some announcements.
Sure. Well, thank you, Carmen, for taking the time out of your very busy schedule for doing this presentation. We really appreciate it. You're the queen of multitasking, like I often say. And thank you, everyone, for participating tonight. Our next presentation will be in November with our new dean of the Faculty of Science and Technology. And that will be with Shauna Zenteno, and she will be talking about viruses. We have a tentative date of November 19th, I believe it is. But we will be confirming that shortly. So watch our website. We will make sure that we post any upcoming events, and also thanks everyone again for participating. Maybe what I will do is, should I temporarily try to unmute everybody if I can?
Actually,
is that a good idea?
Actually, we still have a question. Maybe we take care of that first. Did the researchers who studied the nanohalobacterium actually isolate these guys, or did they just sequence it?
So yeah, they tried really, really hard, Alex, to get them both into culture. The I always get the names wrong. Archaeon. So these aren't quite bacteria. I might have said the wrong thing. They're actually archaea, which are a whole different domain of life. They're really interesting organisms. They kind of look like bacteria, but they're actually more similar to human and plant cells than they are to bacterial cells. So they did get the big one that looked like a sausage. They got that one into culture with just being able to grow it on a chitin enriched medium, but they weren't able to get the nano one into culture. What they were able to do is get them into the co-culture. So they got a pure co-culture. They did a metagenome from that. So they were able to genome sequence the smaller nano one from that. And get that genome. They were able to, from the co-culture, pass it through a filter and just filter out the little ones. and then use that for some experiments. So that was interesting. So they didn't do in that paper, they did a bunch of things. I didn't talk about all of them. They did a whole bunch of growth studies and trying them on different types of complex sugars to see what they really liked to consume. So they did a bunch of stuff, but sequencing was, I just wanted to highlight that for this talk, that they did a bunch. It's a really cool paper.
And there's another question. Isn't it somewhat misleading to suggest that just because organisms can combine for mutual benefit, that they are not still competing with other combinations for survival and dominance?
So are they still competing with each other? Is that what you mean? Is it somewhat, just because organisms can combine, that they're not still competing with other?
I mean that it's not the individual bits that are competing anymore, but it's the combinations of bits that are competing with one another. So it's just sort of escalating the process to larger organisms.
Okay, yeah, are you talking about sort of the Darwinian theory
Well,
of everything?
the other one, that other lady's
right.
statement, eh? That
yeah, no, I think it's actually
symbiosis, but the symbiotic groups are competing with one another.
Sure. No, yeah, you're exactly right. And that you can kind of get a merging of those two philosophies, I think, where, you know, it's whoever can network the best plus outcompete using their networks, right? So I think you can kind of mesh those two ideas somewhat together.
Yeah.
And there's another question from that Adrian. Given the northern boreal and Arctic climes in which lichens populate, is it possible to genetically engineer this triple organism to eat greenhouse gases, especially methane?
Yeah, so this is really interesting. And I don't know, people are more, in this question, people are looking at what they call methanotrophs, which are methane consuming bacteria. So I think for this question, I think that microbiologists are more looking at the bacterial groups that are able to do that, because I don't think that that lichens are, I think they don't think they have the existing machinery quite as readily available as a methanotroph, which is really interesting. And it's a big study, is trying to figure out how methane can get consumed by these bacteria and can turn into something else that's not as harmful. Does that answer your question?
So looks like we have no more questions. I guess we'll leave the screen on for a little bit, and I just want to thank you, Carmen, for doing this. And I believe Linda will at some point post your presentation for those who couldn't make it tonight. We'll post it on the Science Outreach site. And I want to thank everyone for attending, and we are still leaving the Zoom on for a little bit if you have some last minute questions. But if you have to leave, thank you so much and drive safely. Oh no, we don't have to do.
I know.
Compute safely. I note that down on the bottom of the chat, there's 3 little dots. And if you click on those, you notice you can save the chat if you'd like to review it. There's not a lot tonight, but in general, that might be helpful, including the link.
I did that right now. I'm not sure whether it helps.
Well, if you do save chat, it saves the whole chat as a text file.
Oh yeah, I see it.
And notice it says show in folder.
Yeah, that's great.
So,
okay, and I did it too early because there's still things coming in. Thank you messages from everyone. I'll let you read those yourself, Carmen.
Yeah, I think we're good. I think everyone's done asking questions.